All right, so we're going to talk about our last two polymers. I'm going to talk about one briefly because we have a whole unit on it. But I do like to mention it because sometimes people don't realize that it's a biological molecule. So we've talked about two of the biological molecules. What have we talked about? Yeah, carbohydrates and fats. So we're going to talk about the third one. I mean, they're all pretty important.
Protein.
Well, I'm just jumping ahead because we're going to spend all week talking about proteins. I'm just going to talk about these briefly because I just talk about them here, tell you what they're made up of, and then we move on because you have our next unit is all DNA and RNA, and we learn it in detail. Huh? I'm gonna have you guys. I'm gonna teach you how to extract DNA and do some cool like electrophoresis stuff. Yeah, we'll do some.
From strawberries?
No, from you.
Oh.
There's no needles, right?
No needles. We don't do that in here. Do you want to take them out from strawberries? I mean, it's more interesting to take your DNA out, isn't it?
Yeah. No.
Okay.
So
Why does that gross you out?
we could do both.
We could do both. Okay, I could give you a choice. All right. What are nucleic acids? What are the monomers? Do you remember? Begins with the N. U. Yes. Who said that?
Kayla.
Thank you. Nucleotides are our subunits. So when I have put this on your quiz about that has the things that I'm going to ask you about, you're going to have to know all the monomers of everything. This is the monomer of nucleic acids. There are two types of them. What are the two types of nucleic acid?
Diribonucleic acid.
Yeah.
Ribonucleic acid.
DNA, RNA. And their primary function is what?
Instruction.
Yes. It's your genetic info. I just recorded this lecture like 2 days ago, but did I talk about what your genetic info actually codes for?
Protein.
Yeah.
Structures.
Your DNA just has I'm gonna do this. Only has instructions for what proteins are in our body. That's how important proteins are. I mean, DNA is important because it's the instructions. All right, so then we have these little nucleotides. I'm going to draw a picture of the nucleotide. You guys remember me talking about it? It's got a pentose sugar. All right, so what's this guy here? Do you remember? Okay, you need to know this. Phosphate group is up here. DNA actually has a negative charge because it has little O's around. Its negative charge is important for when we are working with DNA later on. This is our pentose sugar. For RNA, it's ribose. For DNA, it's deoxyribose. And this is our nitrogen base. Then I went on to tell you about the 4 different nitrogen bases. We're not going to talk about that much here. This is what you need to know in this unit about nucleic acids. Okay? This is the important stuff right here. The parts. Am I in your way? The middle one says DNA, RNA, and its function is genetic information. Oh, pentose sugar. Pentose. Okay. Now we're really going to get into details. Okay. So a lot of your next quiz is going to be related to what we're talking about this week. There's a lot of stuff in there about protein structure. Okay. So our last nucleic acid that we're going to talk about, and we end up talking about for like the next 2 or 3 weeks, are proteins. I had a medical school professor tell me once that 50% of the time the answer is going to be proteins, right? When you don't know what something is, guess proteins and you're most likely right. And he's been right because proteins do almost everything in biology. So who knows what the name of their monomers are? I hear little mumblings. You're right.
Amino acids.
Have confidence. All right. I'm going to draw the amino acid here, okay? I'm also going to, let's talk about the function quick. Their function, I'm just going to put basically everything. They do everything. And when I brought up that little chart in lecture, I showed you all the different things they do. We talk about enzymes next week for the whole week, and we do this really cool lab where I blend up cow liver and you play with it. It's got enzymes in it. We're gonna play with enzymes. It's awesome.
It sounds delicious.
It is. It's like a smoothie, a protein smoothie. All right.
Drink it.
The structure of an amino acid is something you need to know. So I'm going to do the dehydration synthesis of an amino acid here before I talk about the different levels of protein structure. So, is this in the way? Is it in the way for you? You can move if you need to, okay? All right, so we have, I'm such a dork that I love drawing these. All right. Does this look familiar to you guys? All right. This is the generalized structure of an amino acid. You have to know it. Okay. And what else you have to know about it is the two functional groups. Do you guys remember the last POGL we did? The only POGL we did? There are two different functional groups on this amino acid. Do you recognize these? Yes. Good. Carboxyl. All amino acids have a carboxyl group. Or carboxylic acid, they call it sometimes, right? What is this called? Yeah, amine group. Amino acid, get it? Yes. What's the important thing about the R group? Yes, good. So our R group is important because it's different in every amino acid. This is what makes amino acids different. It gives them their behavior. So if we think back to that first POGL I did that, and I talked to you guys about how we know things. The majority of the things we're going to talk about in here are things that are hydrophobic or not, right? And something is hydrophobic if it doesn't have a charge. If it's something that's polar, it's hydrophilic. Those are the two things that are probably the most important for us, and we spend some time in the next 2 days really learning what ones are and what ones aren't. But I'm just going to stop there. All right. So what I'm going to show you is a question on your next quiz, okay, that you're going to have to know. Are you ready for this? All right, so I'm going to erase this for a minute. So when amino acids join together, right? I'm just going to draw a different color one over here. So sorry if I'm in the way. CCOOH RH. All right. So I have one amino acid here. I have another amino acid here. There are groups. They're both different. That's what's making them different. I have to put these two together. What do I need to get rid of? Yep. Water's going to leave. So we're going to do H2O out. I'm just going to put it like this. HOH out. Right? And then.
Can you just take one OH out?
Nope, you're right. Wait, I have to take the OH out. No, this is it. HOH. So I took the H from the amine group and the OH from here. Okay? Thank you. Normally they are bonds between the oxygens and this bond forms between the carbon and the nitrogen. So what's the name of this bond? Do we remember? And are we getting familiar? Yes. We form a peptide bond between the carboxylic group and the amine group. Now, for bonus points and to be my favorite student of the day, what is the name of this particular structure of a protein? The order of amino acids. Remember we talked about the 4 levels of protein structure? Does that ring a bell?
Primary structure.
Primary structure. Is that what you were going to say, Maddie?
Yeah, that's what I was going to say too.
Okay, you guys are all my favorites for today. All right. You guys are all my favorites all the time. Okay?
That was my answer too.
All right. The primary structure. So we have these different levels of protein structure. So we have levels. How many levels do we have?
4.
Yes. Okay. So today we're really exploring those, and we're going to go into tomorrow because I want to give you enough time to do it. That's how important this is. So the primary structure is the order of amino acids. It's literally what order the amino acids come in. What determines what order the amino acids come in?
DNA.
Yes, good. It's determined by your genetic code. So these are the important things we need to know about primary structure. All right. I'm hoping I can fit it all in here, but I think I'm going to have to erase stuff to do so. Because I got to make sure that we get this. Sorry. All right, here we go. All right, so our primary structure. So anyway, you're going to have a multiple choice question that's going to ask you, like, you guys need to know the bond is between these two guys. All right? You got that one? Put a star on your quiz. All right. So what's the next level of protein structure? Secondary. So who knows what the secondary is? It's kind of vague the way they describe it. Folding or coiling, right? Coiling. That okay, yep, that's an I in there. Pretend. Of the polypeptide. So when we say polypeptide, that just means many peptides. You know that, right, guys? We have alpha helix or beta sheets. Now, I'm going to show you guys something. And I don't know, I think I showed you this in lecture, but we're going to play with it more today. It actually more specifically has to do with the interactions between the backbone of the polypeptide. So the NHs, the H's from the N are going to interact with the O's. So I'm going to write here hydrogen bonds between H and O of the backbone of the amino acids. And we're going to explore that more today. I'm going to put AA here. That means amino acids, okay? So what happens is, as these guys start to get close together, the O's and the H start attracting each other through hydrogen bonds because they do that, right? And they have this secondary structure. Now, the next one is one we're going to be playing around with a lot this week. I don't want to say it's the most important because it's not the most important. Can you guys see okay? Is this in the way? If this just becomes in the way, I can just get rid of it. So tertiary literally means 3. Who can tell me what that is? Go ahead, Alina.
The overall 3D shape of a polypeptide.
Yep. Good. 3D shape. What's it based on the interactions between?
Amino acids and the R groups.
The R groups. Good. So the R groups. The first secondary has to do with this backbone, right? The interactions between the N's, NHs, and the COOHs, right? This has to do with R group interactions. And you guys will be really exploring that too. R group interactions. Good, good. All right. Proteins are really these huge molecules that have these 3-dimensional shape. And when we discuss enzymes next week, you'll understand why the shape is so important. But a protein's function depends on its shape. Okay, so we're going to talk about denaturation in a minute.
Isn't there like an entire thing about the complement system where it's just like in the blood?
Yes, but do we get into that? Not so much. All right, so the last one, not all proteins have this, but what is this one?
For stabilization.
It is for stabilization, but when multiple proteins get together, multiple proteins interact, okay, it's their shape. So I gave an example on lecture about hemoglobin and sickle cell, right? Hemoglobin is in our blood, and it contains 4 polypeptides, 4 proteins, and they all interact to form a shape to carry iron, which carries oxygen. So people with sickle cell, there's a change in their primary, and it goes all the way down to their quaternary and affects them. All right. The last thing we need to talk about before we do start our POGL. What do we need to talk about?
DNA traces.
Yeah, okay. Can I erase this? Drawing amino acids is one of my favorite things to draw. It's weird, I know, but I really like them. All right. Last important thing and what we're going to learn this week. What does it mean to denature? What do I mean when I denature? Huh? Unravel? Good. Change shape. Unravel. Break bonds. If I denature a protein, what have I done to it? Anybody?
Say that again.
If I denature a protein, what do I do to it?
It unravels.
It changes its shape. Oh.
When we denature a protein, we make it useless. It can't work anymore. Protein. I don't even know if I'm getting this part or not. Protein function depends on shape. And I guarantee you'll have an open-ended question on that. Whether on your exam or your quiz, I don't know. Depends on shape. I got to make your quiz not really long, so. Okay. Protein function depends on shape. If its shape changes, its function is useless. Every single protein has a particular shape because it interacts with something else. Okay? So that's really important for us to remember. Oh, some of you guys, do you want to turn in your lipids paper that you did turn in yesterday so I can give you your full grade? I'll take that late. And also, if you forget to turn something in and you're afraid I won't take it late, please always ask. Because the worst case scenario is I say no, and the best case is I say yes, and you guys get the credit you deserve. Okay, so just remember that. Got this all? Okay, bye.